Tracy Johnson. I'll be teaching your lesson today on DNA, both its structure and its function when we talk about the process of DNA replication. The image that you see here at the start of our presentation is of the two gentlemen who are credited with the discovery of DNA's structure. On the left here, this is James Watson, and over here is Francis Crick. And through a very long, very complicated, and a little bit controversial history, these two discovered that DNA is a double helix. If you're interested in more of that backstory and you're curious about all the people who should have gotten credit and didn't, I encourage you to check out the extension activities. So DNA stands for deoxyribonucleic acid, and the name DNA tells you what it's made of. DNA is made of nucleotides, and each nucleotide is made of three pieces. A sugar, ribose, so we've got deoxyribo, a phosphate, which is the chemical formula PO4, and that is linked to the sugar to form the backbone of DNA. By backbone, I'm looking at right here. Phosphate is this little green dot in my diagram. Sugar is this 5-point carbon sugar right here. So these two make the backbone of DNA. And then there are bases. There are 4 bases, adenine, thymine, cytosine, and guanine. Those 4 bases form the ladder or the rungs across the inside of DNA. They are joined together with hydrogen bonds. And I've got a couple quick animations to show you how that works. Let's see if this one will work. So these are what those 4 bases, T, C, and G, look like. On your page, you're just going to write A, C, T, G, but each one of them has a unique chemical composition and a unique shape. As you can see, the A and the G have a 2-ring structure. The C and the T have a single ring structure, and they bond together in that specific hydrogen bond pairing because of those structures and the way that their hydrogen bonds are pointing. When you look at the DNA double helix, this is a 3D model. All of those pieces represent different parts of that molecule. If you take the DNA double helix and you unzip it and make it flat, you can see that both sides are identical. Both sides have sugars and phosphates on the edges of the ladder, and then the rungs of the ladder are made of those A's, T's, C's, and G's. The unique thing is they partner up in a predictable way every time. So if you know what this strand says and you know how they partner up, you'll automatically know what this strand says. Because according to our notes, I'll go back to slide, adenine and thymine are always going to go together. They always want to partner up. They are called complementary base pairs. Cytosine and guanine, same thing. They are also complementary base pairs. Francis Crick, much older in this picture than on the first slide, he's the one who first came up with this idea of a central dogma for biology, a set of governing principles or a standard that all life would follow. And what he came up with is that DNA can make more copies of itself. So you see the arrow pointing back to DNA. DNA can also make a close chemical cousin called RNA, and RNA makes protein. You don't get to skip a step. You don't get to go from DNA to protein. RNA, even though it's simple, does not make DNA in most cases. So this is the standard that we're going to set. This is the rules that most life follows. So the process of DNA replication, or making extra copies of DNA, looks like it should be really simple. Here is our DNA double helix, and as you can see, it's kind of a shadowy animation here, but as you can see, we've got 2 strands. It would be super easy to just unzip each of them, add new little pieces that are surrounding it in the nucleus of the cell, and just keep going until we ran out of room at the top. And that would be a great way to copy. Unfortunately, it does not always work that smoothly in biology, and DNA is no exception. Ideally, this would be the way it would go, but we have some challenges to overcome. There are 3 phases of DNA replication that we're going to go over today: initiation, elongation, and then proofreading. During that initiation step, or the first step, we're going to involve a bunch of key enzymes here. The key enzymes throughout all of DNA replication is helicase. Sorry, something popped up. Helicase, polymerase, and ligase. Helicase is the one that's going to separate the 2 chains of DNA. And a good way to remember it is that helix, so the double helix of DNA, is being separated by helicase. Another way to remember it, and I'm embarrassed that I'm putting this out on the internet, is a cheesy pickup line that biologists like. And that cheesy pickup line is, "Hey girl, if I were an enzyme, I would be DNA helicase so I could unzip your genes." Yeah, pretty bad. Otherwise, you can look at this great picture of Captain America snapping apart a log from the Avengers. So that's helicase. Polymerase, its job is to build a new strand of DNA, and then the ligase is one that joins together or links together any little pieces of DNA that didn't quite connect. We'll talk about that one in a bit. But during that first phase, first thing we have to do is we have to unzip the DNA. I want you to think of, you've never done this, I'm sure, but let's say, for example, you'd written all the answers to a test on your arms, both arms. But if your DNA helicase, or I'm sorry, if you're a DNA molecule, they're all twisted together like this. Can you copy down the answers to the test? No. First of all, you can't see all the answers, and second of all, your hands are tied up. So the very first step is to unwind your hands so that you can now see the answers. Same thing with DNA. In order to see what it needs to copy, we have to unzip it. So we use DNA helicase to do that. We have two strands of DNA, and they run in opposite directions, which makes everything complicated. So if your DNA is tangled up this way, not so bad. Unzip it, you can read them both. But unfortunately, DNA doesn't both point upwards. So I'm trying to see my camera. One strand of DNA points downward. So let's see if I can do this without it tipping over. It's kind of like this. One strand points up, one strand points down, and that twisted up molecule becomes a little bit more complicated. The leading strand is the easy one. We read it left to right. It's just like copying down normal information. But the lagging strand has to make things complicated. It copies from right to left. I'm going to pause for just 1 second. So for example, the leading strand got to copy left to right. Carry On My Wayward Son, fantastic song. Give it a listen today, it'll cheer you up. So if that's the way the leading strand goes, the lagging strand has a challenge. It cannot write left to right like normal. It has to be copied backwards in short chunks. So if this is my starting point, I have to now write carry on. Oh, I'm getting really messy here. My wayward. You guys see how that's messy, awkward? Everything's really choppy. You've got extra spaces. You crashed into work that you'd already written. That's the challenge that we see on that lagging strand in our next step. So when we start adding those new nucleotides to start building the other half, the other strand of DNA after it's been unzipped, we're going to use this enzyme DNA polymerase. That prefix poly means many, so we're adding many little pieces to build the rest of the DNA. We call this semi-conservative because every DNA double helix is going to have one old strand and one new strand that we've just added. We're going to remember that A always partners up with T, C always partners up with G across that DNA double helix. But the way we add those nucleotides is different on the leading and lagging strand. As I showed right behind me here, the leading strand is read continuously, super easy. The lagging strand is written in the opposite direction in these short little chunks. So on my image down here, this would be the leading strand. Easy, keys just gotta go in one direction, everything's fine. The lagging strand is backwards. You're copying in little pieces, you run into stuff you've already done, then you gotta jump back, find another piece, copy that. So I want you to think about, a lot of you are probably playing video games while you're on break. So lagging in a video game or lagging on a video that you're streaming, it's a horrible thing. It causes the video to be all choppy. It causes your video game to be all choppy. Same thing here. We've got a lot of choppy little fragments, and we call those Okazaki fragments. We can't leave them empty like that. We have to link the DNA together, and the enzyme that does that is called DNA ligase. Ligase makes the links. Here's a funny little joke for you. What did DNA ligase say when it was confused? What did it say? I don't understand. I'm lagging behind. Get it? No, that's cheesy. Sorry. So here's an animation of the entire process that we've just talked through. I'm going to narrate it for you. So this is the this is the old DNA double helix we're trying to copy. This blue enzyme right here, that's helicase. As you can see, it's spinning around, breaking those hydrogen bonds, and separating our DNA into two separate strands. This one is our leading strand. Everything's going super smooth. It looks great. We've got a big polymerase enzyme grabbing on right there, and it's just copying that strand. But the weird stuff is happening over here on the top left. That is our lagging strand. As you can see, there's a lot of polymerase enzymes involved. Each one is grabbing a little loop of DNA, copying it, crashing into the work that it's already done, and then it starts over again. So there's another polymerase enzyme, grabs on, crashes into its previous work. So the leading strand, this one, so smooth, everything works well. Lagging strand, not so much. The last phase of DNA replication is called proofreading. And during proofreading, we need to check for errors. So every time DNA polymerase zooms along that strand of old DNA, it It's making sure that I partner up a C and a G, an A and a T every single time. And occasionally it does make errors. Even after all of that proofreading, there are errors that happen. About 1 in every billion letters, which is pretty fantastic. DNA polymerase is pretty good at its job, but when we do have those errors, it sometimes causes mutations. We'll talk about that more later on this week. So I'd like you guys to practice replicating these strands of DNA on your own, remembering that our base pairs are that A and T want to be across from each other on a DNA strand, and C and G want to be across from each other on a DNA strand. So I'll give you just a moment to go through those. 1, GAT, TAC, A. When we partner that up with complementary base pairs, G and C want to be across from each other. So the other side of the DNA double helix is C, T, A. A goes across from this T, A across from this T, C across from this G, and so on. Hopefully you guys got that one right. Next one, ATT becomes TAA on the other side of DNA. ACA, TGT. Let's do one more. This one, CTA, becomes GAT, ATG, TAC, and then A. So by doing this, I've shown you in black what the original strand of DNA was, and then in blue, the new strand of DNA that we created through the process of replication, using those complementary base pairs, A with T and C with G. Good luck, everybody. If you have questions, please make sure that you email your teacher. We miss you all so much, and we wish we were here to do this in person.